did the Jack Weston.
Is this the I only see CP and Psych Soc.
Yeah, I didn't add the B&B because I haven't finished my blind review for the B&B yet. I won't lie.
Oh, but you're supposed to input your scores that you t get before.
Oh, yeah, I was gonna add it after I did, or not the blind review. I haven't finished the complete review. Where's my score thing? But I got 501.
Wait, so
So
what?
I got a 125 on cars and a 125 on B&B. And then I took cars the second practice exam.
When you say the second practice exam,
Yeah, online.
what do you mean?
Like, you know how I finished Chicago and everything?
Oh, you're talking about Jack West.
Yeah, yeah, yeah.
Okay. All right. Yeah, it seems like your score improved.
Yeah, but I also don't know how I did really well on CP because I guessed on a lot of CP. So
Uh-huh.
maybe my educated guesses were well because I don't know how that occurred.
But a score increase of 2 points, I think is too high of an increase to be just due to chance.
Yeah, I don't know what happened. I was like impressed.
Yeah, no. What's happening is you're learning.
Could we actually go over my worst CP passage then? I feel like that's what I wanted to do today because I've done B&B with you,
Yep.
but we haven't done CP yet, and I feel like
Mhm.
that would be useful for me.
Yeah,
If not,
you could do it.
maybe psych would be useful because I had trouble on some of these psych passages, surprisingly. Okay, let me go ahead and look up the passage I did that I wanted to do with you.
Oh, so just to let you know that uh I have to do something from 3:50 to 4 p.m. So for 10 minutes, I'm gonna be like away from my computer.
Mhm.
But yeah, after that, I can come back.
Okay, yeah, that's perfect.
Mhm.
Um, so I wanted to do this one with you. Let me share my screen. Did you want me to open up a new exam or did you want me to open up the review?
The review's fine.
Okay. Okay, let me go ahead and share my screen. So I was hoping we could go over this exam. Oh, sorry. Yeah, this one.
Let's zoom out some more.
Um,
That's fine. Oh,
that's good.
let's zoom out a little bit more.
I feel like it might be beneficial for me to open a new exam because I don't. Let me see if I could go like this. Oh, that could work.
Yeah. So it just some of it's cut off.
Yeah, I don't know if I'll be able to fit all of it.
You could so have it so that nothing's cut off, and then you can go from there.
And,
Like share
oh,
view.
oh,
If you're using like the zoom function of zooming in with so just leave it like normal, but change the zoom from the web browser.
okay.
Like what you're doing now, like you're moving it around, like have it so that it's just regular.
Yeah, I'm changing it on my web browser.
So I mean, change it from what you were using before.
Oh, like zoom in and out, like leave it like this and like go like this.
Yeah, so but just go back
Yeah.
so that you can see the yeah, like you
Okay.
can see it normally. Like go back all the way. Yeah. So this way we get to see the entire thing and nothing's cut off.
Okay. Does it matter that this is cut off? Like the map?
Oh, yeah, that's fine.
Okay.
Um so was this a question of just this was this um
It was like seeing how to read like a passage with
okay. Well,
like
did you get more
like
than one question like wrong on this?
yeah, I got half of it wrong. I got
Okay.
two. It's a four question passage, and I got two of it wrong.
Okay. So so yeah, why don't you I'm curious if you just do one thing, if you so like leaving it the way it is here, but in the browser, if you zoom in on the browser, let's see what happens there. Yeah, you can do that like one more time. Yep, there we go. So you see how it's like you could use the zooming feature with your computer, which
Yeah.
zooms in, but that's zooming in, meaning like when you zoom in, then it cuts stuff off, right?
Yeah.
But if you do it with your browser, it just changes the text size and everything, so you nothing gets cut off.
Hmm.
So, but all right, yeah. Why don't you start by reading it out and when you see like buzzwords, acknowledge them and I'll kind of this seems like it's a good passage to kind of like for me to interject and kind of ask you questions about things as we read through it. So,
Okay.
yep.
The tissue of the brain and eye require glucose to carry out metabolism.
Okay. So, what can you tell me about glucose?
Um, it's part of,
What can you draw it?
do you want me to draw it with on Zoom? I also don't know if I can draw glucose, if I'm being honest.
Yeah. So, um, let's see. So, okay, maybe in the chat I can tell you. I'm trying to think of like so as we go through this, like what to have you like look up and learn to do
Mhm.
rather than just going over because like I'm gonna ask a lot of questions with glucose. So um but let's see how I should do this. So yeah, in the chat I'll just write things that you like things to for you to like look up and get down. So I'm gonna write um like glucose, you know, you should be able to write to draw the straight chain sugar, um the ring forms, both anomers, the epimers for glucose, and know that the brain can only use the for glucose, you should be able to draw the straight chain sugar, um, the ring forms. Um, I think it'll probably be easier if I just write this in the draft an email to you. And
I was gonna just copy and paste it into like my notes on my Mac.
yep. So
So I could just put the meeting on I could just do it side by side and
yeah. So,
dropping things into a notes.
yeah. So yeah, I'll add it to a drafted email so you can then see the email and then see what types of things you need to do. So yeah, you want to be able to draw the straight chain form of glucose, the ring forms. There's two, as the depending on the anomer. You should also know how to draw the the epimers of glucose and also know like how the reaction happens to make that ring
Hmm.
and um and other sugars like galactose, mannose, etc. And oh, and then that the brain can only use glucose and ketone bodies for energy.
Yeah.
So that's pretty much what they're saying here so far. So okay, good.
Okay. This glucose may be obtained through direct ingestion or through breaking down glycogen stores in the liver.
All right. Tell me about this.
So it's telling us how exactly glucose is utilized. So you can either like eat it and get it through a food source, or you can break it down. You can break down like glycogen.
What's glycogen?
Glycogen isn't it a precursor of glucose?
Mm, if it was a precursor of glucose, why would you get glucose by breaking it down?
Um, not a precursor then. Would it be like um I another like another form of glucose, I guess? Like a sto like isn't it like glucose, but it's like when you convert glucose into storage to be saved for later.
Okay. So tell me a little bit more about that.
Like once the body no longer needs glucose, won't you go through what is it? You go through um
So,
like
why would your body not need? So yeah.
you don't need energy anymore. Like you don't need to utilize glucose for energy because you're not like for example, like if you're exercising, then usually you'll break down glucose for energy. But once you finish, then you no longer need the glucose, so then you will store it instead.
So you're giving me a lot of answers that are like bio answers,
Yeah.
but bio answers are just like not really answers. So what I mean, like, so okay, and this is going to be true for the MCAT as well as everything else. Like in bio, they would teach you, okay, glucose is just a sugar, and then you use it, and blah blah blah.
Hmm.
But you want to be able to think of it on that molecular level.
Yeah.
What is like literally what a glucose molecule looks like, right? And literally what a glycogen molecule looks like. So you
Hmm.
want to be so because
So you're
like
kind of learning how each molecule looks like to a T.
yeah, well not necessarily like for each molecule, but you want to be able to you want to have that approach.
Yeah.
So because that's why like if you like I don't know if I probably said this before, but it's I bio is just applied chemistry, chemistry is applied physics, physics is applied math. So
Hmm.
what that means is if you in bio, if you know what glucose is, you
Hmm.
don't know what its chemical structure looks like, and you
Hmm.
don't know the physics behind it as well.
Hmm.
But if you know the chemistry of what, glucose looks like, you can then infer how it behaves biologically. So
Hmm.
you want to be able to be on that level. And out of all of the topics on the MCAT, the most prevalent is biochemistry because it's related to biology, but it's still chemistry, which means it's still there's still things that are happening like that you are expected to know on the molecular level.
Hmm.
So, for instance, glycogen, and like I added this, in the email as well, but glucose can polymerize into glycogen
Hmm.
for storage. And so, you know, again, that's just a bio way of describing it. but you're expected to know more of the chemical ways of describing it. So you want to know that what type of bonds. So glycogen is made up of glucose. So you want to know that there's the alpha 1,4 bonds in glucose. You want to know that there's alpha 1,6 bonds which form branches, and you want to be able to compare that with um like cellulose and amylose and
Lactose.
um, milopectin.
Hmm.
So yeah, so that's going to be really like high yield stuff that you definitely have to know.
Hmm.
So yeah, and just in general also, like I know I'm saying a lot of stuff, but you think interdisciplinarily. Like, so remember that question that we did before, and you were like you didn't think that you were gonna have to do math.
Yeah.
So on the MCAT, like, it's you don't want to think like this is a bio thing. This is a bio passage, so I'm gonna think in a bio way, or this is a chem passage, I'm gonna think in a chem way. Like the ultimate reality is that all of the disciplines, um, are relevant. So you want to be able to kind of see things, yeah, interdisciplinarily. But
Okay.
yeah, okay. So um, I'll just continue reading. So, however, these sources may not always provide adequate energy, and the body may need to synthesize glucose via gluconeogenesis. Okay, what can you tell me about gluconeogenesis?
Gluconeogenesis is the process that occurs in, or I mean, I'm gonna tell. Do you want me to tell you like what it starts with and what it ends with, or
Sure, yeah.
like you start with glycogen, and then when you break it down,
Nope.
or sorry, you start with glucose, and then when you break it down, you end up getting I
So, what do you think genesis means?
oh the creation.
Okay.
So
So, if you're creating glucose, it wouldn't start with glucose, would it?
yeah, I wouldn't start with glucose. You would start with um these pathways. I need to watch your pathway video because I haven't opened, but
Uh,
these pathways really trip me up.
yeah, yeah. They can do that. So basically, and obviously this is all like super high yield, like you definitely have to know this.
Yeah.
So the start off with glycolysis. So glycolysis is the breakdown of glucose ultimately into pyruvate,
Hmm.
and that pyruvate can, if oxygen is present, that pyruvate uh basically can enter into the mitochondria.
Yeah. Oh, and then gluconeogenesis, isn't it? It's just backwards,
So
right?
so for the pyruvate that enters the mitochondria, it then undergoes the Krebs cycle,
Hmm.
which ultimately also all throughout these pathways, which are oxidative, by the way,
Hmm.
uh, means that the electrons get taken by things like NADH, FADH2, and think about those as just electron carriers.
Hmm.
And you make them in glycolysis, you make them in the Krebs cycle, but at the electron transport chain, that's where they ultimately give their electrons away,
Hmm.
complexes, to ultimately go to oxygen, and that's your cellular respiration. Now, if you complete glycolysis and you have your pyruvate, right? Um, and there is no oxygen available, then you start to get lactic acid fermentation. And what happens there is, so glycolysis involves NAD plus turning into NADH, right? So at the end of glycolysis, you should have more NADH than NAD plus, right?
Hmm.
So, but yeah, if there's no oxygen present, you're going to want to continue to do glycolysis. So what can happen is the pyruvate can turn into lactate, and when it does that, it turns NADH to NAD plus, so it replenishes the NAD plus supply that so that glycolysis can still proceed. But then the lactate can go into our blood system, blood supply, um, blood circulation, that is, and go to the liver. And in the liver, gluconeogenesis happens, which can involve the pyruvate or the lactate turning back into pyruvate and then turning back into glucose. So yes, it is glycolysis backwards, but it's a little bit more, it's not that simple because so for instance in glycolysis you have the three irreversible steps.
Hmm.
So how do you go backwards if they're irreversible?
Hmm.
And you'll see that the location of those where those steps are carried out uh play a role.
Hmm.
Uh Basically, because otherwise you'll have a futile cycle.
Yeah.
But yeah, so but I'm gonna add all this stuff
For
to.
someone who wants to master all of these cycles, would you just recommend I like draw it on a whiteboard every single day, or like I'm gonna watch the video you have on it, but what else would you suggest I do? I know it's in Anki too,
Oh,
but I
go to
feel like
the
with
shared PDFs folder.
yes. Oh, it's on my other account because this account is weird.
Yeah.
Let me go ahead. Do you want me to switch to
Hmm.
let me go ahead and
Yeah. So in the PDFs folder, I have a lot of really good notes on it,
I
particularly.
know you have one where it's like the big one with all of them connected, I think, right?
Yeah, yeah, yeah. So, so first, so to answer, yeah, to answer your question, so first, you're gonna kind of do it in multiple passes. So like the first pass could be like understanding like what the purpose is of
Hmm.
the pathway, the location of the pathway,
Hmm.
and then the irreversible steps
Hmm.
and why they're irreversible. And then you can do another pass where you look at it, a little bit more in more detail.
Hmm.
But yeah, I would say to look at so in I have like multiple PDFs there.
Yeah, there's a
Notes
lot.
for oh yeah, do you have it
Oh
on?
yeah. Let me change the share.
Yep.
Share. What's this one? Yes.
Yep. Go to PDFs.
Yes.
Yep. Oh yeah, right there, metabolism OneNote. So so now on the bottom here with the plus sign over
Uh,
here, that lets you zoom in. So yeah, go up.
and just keep zooming. Sorry,
Yep.
let me make the
Zoom
zoom ball.
one more. Zoom in one more time. Okay. And let's go to the top left or just the top. Yeah. So here we go. So I never could find like in textbooks and stuff, they would always have the different pathways written out separately. I never liked that because I wanted to see it all in the same cell, so I drew it like this.
Hmm.
So here we have, and this can give you a big picture view of it all, and you always want a big picture view. So you could see that glucose over starts here and enters into the cell through these transporters, and then you have your glucose here.
Hmm.
And you, I put in this color, teal, regulatory sites that are, spontaneous, therefore irreversible. So you could see in this first step, glucose gets phosphorylated,
Hmm.
and then we have some reversible step, and then we have another phosphorylation of it as the next irreversible step. And then
Hmm.
the last irreversible step is from PEP to pyruvate. Now, if oxygen is present, it'll start to go into the mitochondria where the Krebs cycle happens. However, if it's not present, the pyruvate will turn into lactate. And you can see that in glycolysis, NAD plus is an input or a reactant, right?
Hmm.
So here you can see that NAD plus is being made as a product,
Hmm.
and that can help glycolysis continue to happen. But what also happens is, here we can turn off this or hit this X button here.
Oh, yes.
Yeah. So now what can happen is, so I wrote if no O2, the lactate travels through the blood where it enters the liver, and gluconeogenesis happens where it turns back into pyruvate. I mean, sorry, turns back into glucose.
Hmm.
So now scroll down some. Yep, keep going. So now, like, I wrote this all on a OneNote, which is like a huge canvas, but this is a export to PDF, so it's cut off. But we see here that pyruvate inside of the mitochondrial matrix can turn into oxaloacetate and then leave. So you can go down some more. And so the oxaloacetate can turn into PEP as an irreversible step and keep going. Oh yeah. And then skip this page.
Seems like blank, I think.
Yeah.
Here.
So the PEP over here, and then goes through these steps. And now that first step in glycolysis is the glucose turning into glucose 6 phosphate, right?
Hmm.
So this shows that for the reverse, the glucose 6 phosphate would have to enter into the endoplasmic reticulum
Hmm.
because that's where the glucose 6 phosphatase enzyme is, which removes the phosphate from glucose 6 phosphate to turn it into glucose.
Hmm.
That's so you avoid a futile cycle.
Hmm.
So, so yeah, you want to look at the big picture and you want to look at the irreversible steps and the purpose of it of the pathway and the locations of where the pathway happens, the steps of the
Okay.
pathway.
Yeah.
So, yep. Yeah. And I have a lot of other notes, like handwritten notes too, that go over that stuff with like the purpose and stuff. Like if you can kind of go back to the PDFs folder.
Yeah. Um,
Yeah. So let's scroll down some.
which ones do you recommend I start with, like, kind of getting super familiar with?
Yep, I'll show you. Keep scrolling down. Yep, keep going. Okay, so let's click this one, Metabolism 2. So this can actually show you what I mean by, um, so yeah, let's zoom in.
Sorry.
Oh, yeah, do it with the plus sign. All
One
right,
more.
yep, and let's scroll up. So here I drew two glucose molecules, and just like with two amino acids, and just like with, fatty acid and glycerol molecule, they form a bond between each other through dehydration synthesis. And that's your glycosidic bond, which is like the name of the type of bond, but you also want to know like the chemistry, like functional group for it, which is an acetal. Now, glycogen I drew over here. So you could see that. So I wrote glucose units are linked linearly by alpha 1,4 bonds. So that's like this,
Hmm.
right? So that forms this linear chain.
Hmm.
But the every 8 to 12 units involves a branch, which is formed by an alpha 1,6 glycosidic bond. So here you can see so this is the sixth carbon here, and it can form a bond with the first carbon there
Hmm.
to give you your branch. And if you scroll down a little bit more, this is a macroscopic view of glycogen. So you
Hmm.
can see these linear chains as well as the branches.
Hmm.
And what's the purpose of branching? It increases the surface area, making it more soluble.
Hmm.
So then just one more thing, if you just you can probably hit the right arrow
Hmm.
over here.
I don't think it's. Oh, I thought you meant on my keyboard.
Yep. So now here's some more handwritten notes. So this is what so scroll down. So this is what I mean. So like for instance, pentose phosphate pathway, purpose, right?
Uh-huh.
Generate reducing powers via NADPH and to create 5 carbon sugars, location, cytosol, and then I have various steps with various notes. And if you scroll down, there's going to be stuff for the other
Yeah.
gluconeogenesis. And this is, by the way, the Cori cycle where in the muscle cell, the lactate travels through the blood to go to the liver where gluconeogenesis happens. So yeah, I would say to look over these notes.
Okay. Sounds good.
But yeah, we can go back to that.
Mm, let me share. Okay. Do you want me to keep reading?
Okay, so far we're talking about glucose from eating it, glucose from breaking down glycogen, which is a polymer of it, and that's called glycogenolysis. And then also there could be gluconeogenesis, and now gluconeogenesis starts with non-carbohydrate precursors, so
Mhm.
like alanine and things like that, but not like, any sugar.
Mhm.
So um and then they say that there are several enzymes shared between glycolytic and gluconeogenic pathways, but each metabolic process has its own regulatory steps, and each has enzymes that are unique to that pathway. Much like glycogen storage, gluconeogenesis takes place primarily in the liver. Each of the major macronutrients, fat, protein, carbohydrate, can contribute to molecules that lead to gluconeogenesis. So okay, that's just kind of telling us, about these pathways and stuff. Um, and yeah, I'll continue reading. To begin the process of gluconeogenesis, the liver utilizes pyruvate, glycerol, lactate, and other molecules as substrate. So, you know, so far it's a little weird because here they're saying that carbohydrates can contribute molecules that lead to gluconeogenesis.
Yeah.
But here we have, you know, non-carbohydrates.
Mhm.
So I think what they mean by carbohydrates that contribute molecules could possibly be because every type of biomolecule can be broken down um, into acetyl-CoA.
Mhm.
So maybe that's what they mean by that, but it's weird that they are including carbohydrates in the first paragraph because it's not supposed to include that. But anyway, so we see that these are molecules that can act as substrates for gluconeogenesis. Oh, they're actually explaining a lot here for the pathways. So, this would work if you didn't remember it, like this information. Lactate, for example, is produced by tissues undergoing anaerobic respiration. What's that?
Without oxygen.
Yep. So it's like if you do like some exercise, like where that's not an aerobic exercise, like lifting a weight or something, like eventually it's like impossible to do it because you just build up so much lactate.
Mhm.
Um, and this can be transported to the liver via the systemic circulation. Um, the conversion of lactate to pyruvate is coupled with the reduction of NAD plus to NADH. Um, Pyruvate enters gluconeogenesis after being converted to oxaloacetate. Oh yeah, we kind of just saw this stuff. So
Mhm.
by pyruvate carboxylase, biotin, known as vitamin B7, is an essential cofactor in this conversion. Okay, cool. And now they're going to be talking about fats. So this is a pretty good, like, kind of like a textbook giving us a lot of information. So glycerol, and by the way, you're expected to know how to draw like amino acids, and peptide bonds and glycerol and fatty acids and how that stuff relates to. So, I'm gonna add that to. So, um, because your glycerol molecule basically looks like this. Sorry, these are three OHs.
Mhm.
And when you have your fatty acid, you're going to make a bond, between these to give you your esters.
Yeah.
So, okay. Glycerol produced from the hydrolysis of fats can join gluconeogenesis by after being converted to dihydroxyacetone phosphate by the enzyme glycerol kinase, followed by additional oxidation. Finally, protein can contribute gluconeogenic substrates in the form of glucogenic amino acids. So that's also a thing. There's some ketogenic ones as well. Um, and Such amino acids can be converted to oxaloacetate in the citric acid cycle. Oxaloacetate converted to PEP, which follows a gluconeogenic pathway through PEP carboxykinase. Okay. Anything below?
The pathway.
Oh, nice. So yeah, they even draw it out for us, which is good. So, yep. So we have glucose. Oh wait, in the pathway of gluconeogenesis. Okay, glucose and oh, so can you like it doesn't let you scroll down on the left anymore?
No, it doesn't. I could try zooming out.
I don't know. It's not a zooming issue. I think it's just the scrolling thing. But I think up here you used to have something that you can drag up and down for like your highlighting versus.
Oh yeah. Let me see if I moved it completely out of the way. I can also try to open it up completely. I know we drove on this or wrote on the screen though.
Oh yeah. When you say open it up completely,
Like, well, I guess it is opened up completely. It's just zoomed out a ton.
oh, you mean like with the background? It seems like it just open.
Yeah, but I think it's because I zoomed it out so much. That's why it's like this. Because I have it open on like the full screen thing.
Uh, it's weird.
I could also just close and reopen it if you want.
Uh, yeah, we can do that.
Okay.
Um, so yes, I'm gonna have to do
Yeah,
something now. I'll be back in 10 minutes, okay?
yeah, I can do that while you're gone and fix
All right.
it.
Thanks.
Okay, let me go ahead. I think I figured it out.
Mhm.
Um, right here.
Okay.
Doesn't fit the caption, but this is the best I could get it. Oh.
So we could see that, oh, they give us entire. Oh, this is cool. They give you the entire gluconeogenic. Uh, so we can see that there's multiple entry points. So like you can go from the pyruvate, but you can also go from the glycerol as well, right? Right over here.
Mhm.
All right, cool. Anything below that?
There's a caption.
Oh, okay.
Or the caption, then what they put.
Got it. All right.
Yeah.
Any questions so far?
No, just realizing that there's a lot of, like you're right, interdisciplinary biochem combining with chem. So
Hmm.
maybe that's why I struggled with this passage. I feel like my biochem review hasn't been as strong.
Yep, got it. Got it. So let's see. A certain metabolic process in the liver produces NADH as part of the process. If this process is upregulated, which of the following effects associated with gluconeogenesis is most likely to occur? Okay.
I'm assuming I would look at the image for a question like this, right?
Well, the image doesn't show you anything about NADH,
Yeah.
right? So
Mhm.
we want to figure out what metabolic process, you know, they're talking about that creates NADH, and that is this is happening in the liver.
Mhm.
So, what are your thoughts?
Um, so I mean, do you want me to start with like I'm pretty sure I guessed on this one, but I'm
Let, let,
like
let's try this. Let's try thinking about it first. Um, okay, what is this like? Kind of like describe like tell me what is this question asking us?
so what it's a kind of like okay, um there's a process that is going to produce NADH. If we increase the amount of this process occurring, then how does that affect gluconeogenesis?
Okay.
So,
So, yeah,
um, then I would have to think, okay, what is the role of NADH?
okay.
And NADH, when I think of NADH, I think of electron transport chain instantly.
Tell me, uh, tell me more. What do you mean?
Um, how NADH is an electron carrier.
Okay. So, if something makes NADH, so we can look through the passage to see what they say about NADH, right?
Mhm.
So,
Oh, NADH. Yeah, I was gonna say the reduction.
okay. So they're saying that um, if you turn NAD plus into NADH, that can contribute to um okay, sorry. So they're saying that this NADH that's getting produced um in the context of gluconeogenesis, right?
Mhm.
Because they're saying conversion of lactate to pyruvate is coupled with a reduction of NAD plus to NADH.
Mhm.
Then that pyruvate enters gluconeogenesis, right? So we can say that gluconeogenesis can use up NAD or sorry, can produce NADH.
So,
So, okay, let's actually here, let me these choices. All right. So they're saying that so I believe they're saying essentially that gluconeogenesis um produces the NADH, right?
mhm,
Okay. Conversion of lactate to pyruvate is coupled with a reduction of NAD plus to NADH. We can't assume that this is the process being discussed. All that we're taking away is that we're making a large amount of NADH.
yeah,
And if it's happened, then further conversion of NAD plus to NADH will be suppressed. Therefore, the production of OAA and pyruvate will be decreased since pyruvate production coupled with the conversion of NAD plus to NADH.
yeah.
I don't think they mentioned. Okay. Okay. So in uh okay. Okay. So I guess they're telling us that the conversion of lactate to pyruvate it involves producing NADH,
Mhm.
and that's something that can enter gluconeogenesis. So we can say that gluconeogenesis would cause the production of NADH. But if we already have a large amount of NADH,
And gluconeogenesis decreases.
yeah, because gluconeogenesis makes NADH as a product. So if you have too much product of NADH, Le Chatelier's principle will shift that reaction to the left.
Okay.
Now, for these other choices, let's see. A says intracellular levels of oxaloacetate will increase. So that can happen, in the step from pyruvate to oxaloacetate for it to leave the. Oh yeah, they have it here. Yeah. So okay, so yeah, oxaloacetate is in that process of gluconeogenesis,
Mhm.
and let's see. Okay, plasma glucose. So we're not really talking about whether or not glucose is in the plasma or not. I mean, we're talking about within the cell, the metabolic processes within that. So yeah, so that should rule out B and D because those are happening outside of the cell.
Mhm.
So here, yeah, so I guess here the way to approach this would have been is gluconeogenesis producing NADH or uh producing NAD plus, and I think they told us in the passage, at
Mhm.
least for the pyruvate to or the lactate to pyruvate step, you are creating NADH, and then they say that pyruvate enters gluconeogenesis. So we could say that gluconeogenesis produces NADH. So if you have too much NADH to begin with, it's going to shift the reaction to the left, meaning less gluconeogenesis.
Okay. Wait, yeah, that makes sense.
Mhm.
Okay.
Yep. Sorry, I just went to get some coffee. Um, Conversion of pep. Oh yeah, so like yeah, like I said, like it's important to know the locations of these metabolic processes. So conversion of pyruvate into glucose requires pep carboxy kinase, and this enzyme is present in. So I in that drawing or the pathway that I showed you that I drew,
Mhm.
you saw um that deoxaloacetate with or deoxaloacetate within the mitochondria uh, can turn into malate and aspartate and leave the mitochondria, where the oxaloacetate will turn into pyruvate, or rather, oxaloacetate turns into PEP, and PEP eventually turns into pyruvate.
Mhm.
So they're just asking where is this happening? And it can't happen in the mitochondria because the mitochondria is just where pyruvate can turn into oxaloacetate. But there's no PEP happening in the mitochondria. Uh, but the enzyme could be in both. Let's see. PEP CK, they said conversion of pyruvate to glucose requires an initial conversion into oxaloacetate in a reaction catalyzed by pyruvate carboxylase in the mitochondria. Um, Oxaloacetate is then decarboxylated and phosphorylated by cytosolic or mitochondrial forms of PEP-CK after transport of either oxaloacetate in the form of malate or PEP directly from the mitochondria. The remainder of gluconeogenesis takes place in the cytosol. Okay, that's weird. Because yeah, I would have thought in the cytosol only.
Mhm.
Um, but apparently there could also be okay, I guess apparently there's also, I guess PEP CK in the mitochondria. So I don't really see anything in the passage that would indicate that.
Yeah.
So tough one. Uh, so yeah, I would just, add that to your list of notes or Anki cards or whatever that PEP
Mhm.
CK can be present in both the mitochondria and the cytosol, yeah.
Okay. Sorry. And mitochondria. And Sorry, I just wanted to write it down so I remember to make an Anki card.
Mm-hmm.
And then I knew this one because I feel like I've seen this table a lot, and so I don't really need to review this one. And then this one. Oh, I feel like I don't remember if I knew this one or not, or if I guessed on it and somehow was right. Yeah, I feel like I definitely guessed on this one.
Okay. Conversion between glucose and pyruvate strongly favors the formation of pyruvate, and yet the gluconeogenic pathway is able to utilize several shared enzymes with glycolysis to create glucose from pyruvate. It is able to do this primarily because, so typically, um, if you have, so first of all, they're talking about the reversible reactions
Hmm.
because if they're talking about the irreversible reactions, then the answer would be that the enzymes are different between glycolysis and gluconeogenesis, and that can be in a specific location.
Hmm.
But since they're saying shared enzymes, that must mean the enzymes in the reversible steps. Uh,
Hmm.
and when you have a revert like A turning, into B and it's reversible, um, you can have B turn back into A simply if you have a higher concentration of it for it to be Le Chatelier, essentially.
Hmm.
So the formation of glucose, fructose 6 finase, and PEP through so that includes what I said about these are the ones that are just present for gluconeogenesis. So these are not the shared enzymes, right? So that's that.
Hmm.
Push the equilibrium of reactions catalyzed using shared enzymes to favor gluconeogenesis. So the these irreversible steps produce products that increase the amount of like so if this is like glucose and this is like pyruvate, it's like if you increase the amount of pyruvate, you can drive that reaction backwards.
Hmm.
So yeah, it'll be that one.
Okay. And then, so that's all of that. But I'm wondering if there's another passage on here that I can review since we have some time.
Mhm.
I know there's one more that like really tripped me up. I don't think there's this one. Oh, I think it was. It wasn't this. Mm-mm. I don't remember what passage it was,
Okay.
but if you wanted to review, I feel like this one might be a good one to review because this was something I did like I understood it after, but I remember thinking I thought impurities would increase the boiling point. I think this was the question. Yeah. Significant impurities were detected I thought if there were impurities, at least I was thinking back to when I took OChem lab, I thought the melting point would increase.
You're thinking of boiling point elevation,
Yeah.
but so what are the other colligative properties?
So there's boiling point, melting point,
Well, I mean like boiling point what?
elevation and depression.
What do you mean depression?
Like,
You're saying so I mean like for each of them, tell me.
what do you mean?
So like boiling point elevation is one. What's
Yeah.
the others?
There's um just boiling point depression, not one. I swear I thought that was one.
Well, how could you have boiling point elevation and boiling point
Maybe
depression?
it's melting point depression then.
Freezing point depression?
Yeah, freezing point depression. Sorry, I can't remember them at the top of my head.
Yeah. So I'll add that to the drafted email
Yeah.
because that's really important.
Yeah.
Because so basically, yeah, you'll have your boiling point elevation, you'll have your freezing point depression, and freezing point is the same thing as melting point,
Mhm.
right? Just in a different direction.
Mhm.
So you want your melting point to be smaller.
Yeah.
So apparently they're saying the passage says that the melting point of pure caffeine is 235 Celsius.
Yeah, it says it somewhere. I don't remember where,
Huh?
but I remember reading it.
Yep.
Oh yeah, melting point right up here. Yeah.
Yep.
230.
So our answer must be smaller than,
Okay.
than 235.
Okay, I guess I was just mixing up the colligative property.
Hmm.
And then this was another one I missed.
Okay. Methods used in the experiment. Let's look at the experiment.
And I guess it's because IR spectroscopy, and I know there aren't questions on it, but I remember it talked about spectroscopy in this. But stuff like IR and all of like those methods like really trip me up. Like I understand TLC kind of. It's like the polar stays to the bottom, and then the non-polar will typically rise given the solvent front. But it's like all the rest, I have a very like broad, I guess, knowledge on like understanding like all of the methods. So I don't really understand how deeply I have to know like each um what is it of like the kind of methods because sometimes there'll be questions like I remember I think it was this exam there was a question where it was like oh there is a peak at 3020 on like the IR spectrum like what does this
That'll be
mean.
like alcohol, right?
Yeah. And so I think I put that there it meant there would I think the answer I got it wrong. I think I put that that meant there was a carbonyl.
That'll be 1700.
Yeah. So I guess I need to like memorize all of that stuff, but then I don't like I guess like I don't understand how much I need to grasp or what I need to grasp or
For
like with each of these like methods. So I feel like it would be good for me to kind of like review them, but I'm not sure like where to begin. Kind of like I guess like with like the metabolic pathways, like you said, start broad, like start with like the
so
location.
so we're talking about two things here. So we're talking about one, which is spectroscopy, and because spec means light,
Yeah,
so spectroscopy that includes IR, it includes UV vis, it includes mass spectroscopy,
yeah,
spectrometry, I mean, um, and things like that. So I put that in the email, and then also, so then the chromatography is under the category of separation techniques.
mhm,
And in the shared drive, in the shared spreadsheet, I have like, the tabs for like the content. Like,
Yeah, I saw that. That's also something I've been meaning to ask you how to use, like, or if I should just go through and
so
check
that's
things.
so that's literally from the AMC content outline. I just turned it into a spreadsheet. So it's if you wanted to see if this is fair game on the MCAT, that'll show you like literally like, what you need to know and stuff. So for all of the things that I'm putting in this email to you, go to that
Spreadsheet.
those tabs, yeah, and then you can hit like Control F or whatever.
Mhm.
Yeah, you can hit like Control F or whatever the Mac equivalent is of Control F. Yeah, and like type in let's type in like SPEC. Um oh, maybe a T after. Uh oh yeah, right underneath, scroll down a bit.
Yeah.
So yeah, so you see how there's infrared region, there's NMR spectroscopy, um, and there's probably a bunch of other stuff. Let's see.
Oh yeah, mass absorption and emission line spectrum.
Yep. So for the list of topics that I'm sending you, you can go here first and then, look at, the thing for it, and then you can like learn about it and do practice problems on it. And then you can come back and then you can change. See the separation techniques down here
Mm,
on the bottom left? Separations and purification here.
yeah. Okay.
So you see distillation, you want to know. You want to know chromatography, all the different types. So as you review stuff, you can use this to see like what exactly you need to know.
Mm,
And then you can update the drop down menu from like weak to okay. Like right
mm.
now for these, I would put like for weak, but yeah, you can change it from weak to okay, as you learn stuff.
Yeah, because I have never updated. This is, I guess, what like originally was in the spreadsheet. I haven't touched this myself.
Yep, yep, yep.
Okay, sounds good. I can go through that then.
Yeah.
Um, in terms of like, I think I'm for today, I don't like have unnecessarily anything else that I kind of thought to go over. I just thought that passage was going to take a lot longer. Uh,
Uh-huh.
So if you want, like, I can just pay you and then I can like get back to finishing stuff, or I can do something else. It's up to you.
Uh, yeah, we can do that.
Because I think I have a lot of, well, now I know I have a lot of random things to review.
Mhm.
Sorry, do you suggest for this list that I have now in the email draft, should I just watch YouTube videos or like what do you suggest? Because I know there's UWorld stuff, but I think we talked about how last time UWorld doesn't have like like it's like it has like the overall like subject, but it doesn't have like the little breakdown.
Yeah.
Or should I go back to the Kaplan books?
So, um, so interestingly enough, so like, uh, I know like people of like your generation prefer like video and stuff. Uh,
Yeah.
but like with one of my other students, like I realized, I don't know if I've ever shown you like that soft skills paper
Yeah,
that,
you have. You have. It's
yeah. So in it, it says processing information while reading is a skill which can take years to develop.
yeah,
The
we talked about this last time, I think.
Yeah, the best way to strengthen this skill is by simply reading as much as possible. So, I mean, for me, like studying for it, like, I would read like I would essentially have like I have like more than one monitor, but I would have like an MCAT book on one, a textbook on the other, and like Google like image search on the other one. And then when I would take my notes, it would be like cross-referencing everything.
Huh.
And that makes it super fast because reading is way, 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 way faster than like watching a video and stuff.
Yeah.
There's that, but more importantly, since the MCAT is a reading test, you might as well get used to reading because another thing that I noticed with like students that are not, you know, into reading is that reading something takes a lot of effort for them.
Yeah.
And, you know, and if you're someone who is used to reading, it like for me, like it doesn't take like effort to like really read. Um, and yeah, and for the longest time, I was just like, okay, it just this generation is more video oriented. Uh,
Mhm.
but then I kind of realized that this test is not very, video oriented. This test is all
Reading.
about reading. And like that's also why humanities majors score so high, especially higher than, bio majors on the bio like BB section.
Yeah.
Mentioned that before, right?
Yeah, you have. Think I have all my old college textbooks, so I can pull those out and
So what I would recommend is so in the shared drive,
yes.
there's like I have some really good textbooks in the textbooks folder, like a biochem book from Leninger, a really good anatomy physiology book from Saladin, uh,
Yeah.
and some other stuff. That
Yeah.
can be you can look at those textbooks, but I also have a lot of like MCAT books, and in the TBR folder, it's the Berkeley Review. And
Uh-huh.
out of all of like the MCAT books, like this is definitely the best.
Okay, because I was gonna say I've read the Kaplan books already
Yep, yep.
twice, and I don't feel like they work for me at
Yeah,
least.
yeah. So you want to give the here. So yeah, so for instance, yeah, in the Shane MCAT folder, go to the
Yeah.
TBR here.
Yeah.
And you can click on so like let's see, like organic chemistry. Um, we have oh, this is pretty good because it goes over the biomolecules and so like let's type in distillation. So that's going to be so like this is section 8, and you can see separation techniques, distillation, vapor pressure, basically all of that chromatography, extraction, right? Basically all of the stuff that we just talked about, right? And then it starts off with like these goals, like 6 goals here. Like this is what you want to come out of this chapter with, understanding right? The different forms of distillation and stuff. And, then, if you read it, they explain things and with some detail, but excellent, like, detail. Like I wouldn't get too bogged up in the details though, but as long as you can come out of it with those section goals that they show in the first page. And then at the end of each, they also have questions within the chapter, but at the end of each chapter, you get key points like
Mhm.
distillation, chromatography, etc.
Mhm.
And then you have passages. And the passages are quite good as well, and
Oh,
the
nice.
explanations are really good as well. So I would say that so yeah, I would say normally in the past, in the very recent past, I just accepted that like younger students prefer videos,
Mhm.
but recently I realized this test is all about reading. So, and according to the paper, the published paper, right? The only way to really, or the best way to strengthen the skill of processing information while reading is by reading as much as possible. So
Mhm.
might as well start that now, right?
Yeah.
So
Okay.
I would
I will
recommend,
look into that. Yeah.
yeah, so I would recommend using so yeah, you'll see the email and then you can use the content spreadsheet tabs to mark things off like week, your level of understanding, and then you can go. I would say to start with like the Berkeley Review book.
Mhm.
You could also use. the textbooks. You could also use Anki, and of course, I'm not saying you can't use videos. Like if that helps, whatever, go for it, go for it. Um,
Okay.
but yeah, so that's pretty much what I would say.
Okay. Sounds good.
Yep. So aside from that, it seems like you're improving. Uh,
Yeah, I was so happy with my 126. After I saw that, I was like, okay, I'm in shock.
Yep, yep. So yeah, we are on the right track. Yeah, your cars went up as
Yeah,
well.
my cars also went up.
And
But
you're
I'm
wait,
wondering if it's because I like
you're
slow key read out loud. Like I didn't read super loudly, but I read out loud and I feel like I like did it exactly how we do it in our like videos, I guess.
yep, yep.
And I'm wondering if that helped me like I guess like kind of digest the information a little better, which I know I can't really do on the MCAT, but
No, that's exactly what you did because you're learning then because a lot of people they start off with I read like separating reading and understanding. It's like a lot of people start that way. It's like, all right, I just read the sentence and now I'm gonna think, what did that mean?
Mhm.
Right? But when you're going slowly, like the way that you just described, you're understanding as you read.
Mhm.
So it's like instead of reading it fast and then taking time to understand it, you read it slowly, but you understand it at the same time. So it seems
Yeah.
like your score went up everywhere except for Psych Soc, but like
Yeah.
Psych
Yeah.
Soc is a content thing. That's not
Yeah.
I wouldn't worry
And I
about that.
also noticed there was like a 1 point between me and the 126 range.
Yeah.
But and also I think I went super fast this one. I finished it in 60 minutes instead of the full 90.
Yeah, that's the thing with Psych Soc. It's like it's typically the section that people have like a lot of time left over, but since it's so content based, it's not like you could use that extra time to like figure stuff out, right?
Yeah.
People usually are like, oh my God, like, like I remember like even when I would get to that section, I'd have like butterflies in my stomach because it's like, oh my God, it's almost over. And you feel like you want to leave, but you just want to stay and just kind of make sure. But yeah, no, your scores improved pretty much everywhere. Your CP went up, your cars went up, your BB went up, so you're doing an excellent job. And yeah, once you learn that stuff, you can, I guess you'll be taking your next test and then you can learn from that. So,
Yeah. My next test I'm planning to take Tuesday, the
uh,
24th. I'm
got
taking my next test Tuesday and then I'm doing my review on the 25th because
got
I
it.
have off work both those days, so it seems convenient.
Okay. Awesome. Awesome. And I guess by that point you will have finished your blind review and review.
Yeah, I'm going kind of slow on the biochem because I am seeing a lot of content gaps. I feel like I'm better at the reading, but there's just so many
Yep.
content gaps, I feel like, and I feel like it's so dense.
hmm Got it. So, okay. Awesome. Awesome. And yeah, of course you could also use like the PDFs from my
Mhm.
thing and my S tier study creation, like,
Yeah, I think I need to delve into this drive
Mhm.
and really start utilizing it instead of trying to tackle it all on my own.
Yeah, yeah, yeah. So yeah, you're yeah, you're doing a great job. You're on the right track. So awesome. So let's see for today, subtracting 10 minutes, so that's just gonna be 1.5 uh hours. So 113 for today.
I sent that over. There you go.
Awesome. So have a good weekend, and I'm seeing you.
I think I made an appointment for every Friday before, except wait, no, not the 27th. I made one for the
Yeah, okay, yeah. Saturday the 28th.
28th.
All
Yeah,
right.
and I'm going for the second after that.
All right, I'll see you then, and have a good weekend and holidays, and good luck on that. Bullying. So, good work.
Okay, thanks, Shane. Bye.